Low tryptophan, genes turn on. High tryptophan, genes turn off. But how does the cell know when to flip the switch? I'm Miss Parrott, and in this video, we're going to be getting deep into the Trp Operon function, how the cells know when to turn genes on and off, and how they actually do it. So remember from the previous video about the Trp Operon structure, check it out up here. The Trp Operon is made of a promoter, where RNA polymerase binds, and an operator, which is where the repressor protein binds, and then 5 structural genes that will make the polypeptides needed to actually make the amino acid tryptophan, which is necessary for the building of all proteins. The goal of all operons, including the Trp Operon, is to save the cell energy. So all these genes produce polypeptides that are all part of the same process. And so it has condensed them into an operon to save energy. So it will be turned on when tryptophan is absent in the gut. And then it will be turned off when tryptophan is present in the gut. Because if it's present, they don't need to be making it. So, Trp Operon is considered a repressible operon. It's usually on, making tryptophan, again, an essential amino acid needed for the production of all proteins. But it is repressible, meaning it's turn-offable. So it's usually always on, but it can be turned off. We're going to contrast that with the lac operon that is inducible, meaning it's usually off, but it can be turned on. Now, the default position, like I said, is that there's no tryptophan in the gut, no or low, and so it's on. If it's on, then that means the RNA polymerase is binding to the promoter and going past the operator gate and transcribing all of these into one long mRNA. Then, and in this case, the repressor is inactive. And so if the repressor is inactive, it can't bind to the operator. So we're just going to transcribe and transcribe and translate and make the polypeptides needed to make tryptophan. So transcription will occur and Trp tryptophan will be produced. That's the default. But what happens when either I eat a bunch of tryptophan? So say I have a turkey leg or a chicken leg, or I have some dairy or some pumpkin seeds, some food that's full of tryptophan. Or let's say I've been making a bunch of tryptophan and I don't need to make it anymore. Well, if tryptophan is high, the operon will turn off. How does this happen? So the tryptophan amino acid itself is what's called a corepressor. A corepressor will activate the repressor protein by attaching to it and changing its shape. Now, the shape of the repressor-corepressor combo is such that it can now attach to the operator and block RNA polymerase. So RNA polymerase can still attach to the promoter, but it cannot get past the repressor-corepressor complex that is now blocking the structural genes. So no tryptophan will be produced and will save energy. Again, because we already have high tryptophan, we don't need to waste energy making more. The Trp Operon is a classic example of a negative feedback mechanism. This is a huge thing for cell communication in general. So a negative feedback loop is one in which the product of the pathway will actually go back and inhibit the pathway. So This exists to conserve energy and maintain homeostasis in the system. So let's look, this is a diagram that you might see on an AP Bio exam. So if you have no tryptophan, right, then the cell needs to make it. So the Trp operon will be on, will be making tryptophan, but then when that tryptophan builds up and we have excess tryptophan, that is going to activate the repressor protein. The repressor protein will go to the operator, and the Trp corepressor plus the repressor protein will turn the Trp operon from on to off. So just to wrap up, the Trp operon is usually on, but it's repressible. It can be turned off. The repressor is activated by the corepressor, tryptophan itself. And like I said, tryptophan is what we would call a corepressor. So you need them to be a squad to actually repress at the operator. Coming up next is a video about how the Trp operon might show up on the AP Bio exam. Remember, that's coming up on May 4th. So if you haven't already, please check out the AP Bio exam skills series for all of my tips on how to take all that you know and actually apply it in the way you're going to see it on the AP Bio exam. If you found this video helpful, please do give it a like, subscribe to my channel, leave a comment, a question, or even just an emoji down below. And if you want some practice resources for FRQs for any of the AP Bio math and statistics stuff, please check out my Teachers Pay Teachers resources at Know and Grow with Gnomes. Keep learning, keep growing, and we'll see you next time.